Recalling a standard, sad memory, a breakup or a loss, is painful, but the event feels localized to the past, a closed chapter drifting into history. Recall for someone with PTSD bypasses this sense of history. Recounting the trauma triggers an intrusive, present-tense reality. For decades, the medical community operated on the assumption that PTSD was simply a standard bad memory that the brain had amplified to an extreme degree. A functional MRI study from researchers at the Icahn School of Medicine at Mount Sinai and Yale University recently overturned that assumption. The scanner data revealed a departure from traditional memory processing. The brain treats these traumatic events as an entirely different cognitive entity. The research team placed participants diagnosed with PTSD inside an fMRI scanner to listen to deeply personal audio scripts, one narrating a sad autobiographical memory and the other recounting their specific traumatic experience. When listening to sad narratives, patients' brains reacted as expected. The hippocampus, the memory center, actively lit up. But listening to traumatic narratives, the hippocampus remained dark. Instead, the trauma data activated a different region, the posterior cingulate cortex, or PCC. While the hippocampus failed to find common ground between different traumatic stories, the PCC showed shared, predictable patterns across the group. This region functions as a cognitive bridge, linking external events directly to an individual's sense of self, rather than sorting them by date and time. Understanding how the brain misfiles trauma into the PCC requires an examination of the biological design of the human stress response. The evolutionary purpose of memory is survival. Retaining intricately detailed information about a threat ensures you know exactly how to avoid it the next time. When a stressful event occurs, the fight or flight response activates the amygdala. This small almond-shaped cluster immediately floods your system with cortisol. This cortisol flood acts as an emotional tag, a chemical marker that flags the specific experience as essential data for the brain to keep. This chart shows the relationship between stress and memory. As stress levels rise along the x-axis, memory performance improves, up to a distinct peak. But if the stress continues to rise past that optimal point, memory consolidation drops sharply, forming an inverted U-curve. During a sudden, moderate crisis, that peak tagging mechanism creates intensely vivid flashbulb memories. However, chemical tagging is only the first step. To make that data a permanent part of your past, the brain must go offline. Permanent storage happens at night through a biological process called selective consolidation. Rapid eye movement, or REM sleep, provides the precise neurobiological environment for this work. Distinct theta oscillations pair with shifting levels of the neurotransmitter acetylcholine to create optimal conditions for memory processing. As you sleep, the brain actively seeks out the events you emotionally tagged with cortisol during the day and spontaneously replays them. This replay process is pointedly targeted. The brain deliberately strips away neutral background details to prioritize the core emotional threat. Firing these prefrontal limbic connections over and over wires the threat into long-term memory. This biological sorting system is optimized for dealing with an isolated threat, but it breaks down when stress becomes continuous and inescapable. Chronic stress occurs when the body never returns to a state of homeostasis. The fight or flight response stays on, and stress hormones remain elevated. Prolonged exposure to high levels of cortisol is toxic to the brain's architecture. Historical studies on Vietnam War veterans first revealed that this sustained chemical exposure causes the hippocampus to physically shrink. This diagram compares a healthy hippocampus to the structural damage caused by chronic stress. Veterans with PTSD exhibited an 8% reduction in right hippocampal volume, while patients who survived child abuse showed a 12% reduction on the left side. With the primary memory center physically compromised, the brain can no longer route the heavily tagged trauma away into the past. The overloaded neural data is routed instead to the posterior cingulate cortex. Because the PCC focuses on immediate self-representation, the event's semantic structure degrades. It cannot become a standard, time-stamped memory, leaving the person trapped in a raw, intrusive present-tense loop. The inability to move past trauma is a quantifiable physiological misfiling. This structural reality takes PTSD out of the realm of an abstract emotional burden and defines it as a precise neurological target. Future clinical interventions can now aim directly at this misplaced data, attempting to migrate these rogue cognitive entities out of the PCC and successfully route them back into the hippocampus. This process relies on memory reconsolidation. When a traumatic event is brought to conscious awareness in a safe environment, the neural data unlocks, becoming susceptible to alteration. Emerging techniques like targeted memory reactivation during sleep could eventually act as the bridge to facilitate this neural migration. Our evolutionary mechanisms can trap us in our darkest moments, but mapping this biology provides the necessary coordinates to finally lay the past to rest.